Today we'll learn about extracting genomic DNA by spin column method. Here we have taken QAM DNA mini kit. In a microcentrifuge tube, 20 microliter of proteinase K solution is pipetted and to it 200 microliter whole blood sample was added. It was thoroughly mixed and to it 200 microliter of lysis buffer was added. This was gently tapped and mixed well. After that, it was put in vortex mixer for thorough action, and that would lyse the cell membrane and nuclear membranes. It was incubated for 10 minutes to take the lysing process to completion. Then briefly centrifuged to remove drops from the lid. 200 microliter of ethanol was added and mixed thoroughly by vortexing to remove any lipid components. Briefly, it was centrifuged again to remove droplets from the lid. Now the mixture was pipetted into a mini spin column of 2 ml collection tube and was centrifuged at 8000 rpm for 1 minute. The flow through was discarded along with the collection tube. Care was taken to put the solution exactly at the center of the mini spin column. The mini spin column was placed in a new 2 ml collection tube and 500 microliter of wash buffer 1 AW1 was added to it, centrifuged at 8000 rpm for 1 minute. And then with wash buffer solution AW2, the same process repeated and centrifuged at 14,000 rpm for 3 minutes. The flow through was discarded and now the spin column is taken in a microcentrifuge tube and centrifuged at full speed at 14,000 rpm for 1 minute to remove the wash buffer carryover. Now the spin column is placed in a new 1.5 ml microcentrifuge tube and 100 to 200 microliter of elution buffer AE was added, incubated at room temperature for 1 minute. Centrifugation was again done at 8000 rpm for 1 minute to elute the DNA, and this can be preserved at -20 degrees Celsius for later use. Now for the agarose gel electrophoresis, where we can see how much DNA we have actually eluted. For this, we take molecular grade 2 gram of agarose powder, which is mixed with 100 ml of 1x TA buffer. It was microwaved for 1 to 3 minutes till it was completely dissolved. Then it was cooled to 50 degrees Celsius and then 2 to 3 microliter of ethidium bromide was added. Ethidium bromide helps visualization of DNA under UV rays. Now agarose gel was poured on casting tray with the comb in place. The gel was allowed to set at 4 degrees Celsius for 10 to 15 minutes and then the combs were removed and kept in the tank. Now the electrophoresis buffer tanks were filled with 1x TA buffer till the gel was covered. DNA ladder sequence and DNA extract, they were mixed on a polythene sheet, taking 4 microliter of DNA extract and a loading dye of 2 microliter, and then this total amount of 6 microliter was charged into the wells. The DNA ladder sequence was put on the first well. Now the electrophoresis tank was covered and it was run in 8200 volt for 20 minutes. Hardly it will move up to 1 inch distance, and then it was removed and it was visualized in the UV trans illuminator. With the standard sequence, the amount of DNA eluted is evaluated, and thus the process of genomic DNA extract comes to an end. Thank you.